Okay, crew, so I have to speak a little bit quietly because I'm in the library doing this. Shh. In fact, I'm in one of those study rooms where it says, don't speak. So I might get caught and kicked out of here in a moment. So let's first talk about the difference that exists between point mutations versus chromosomal abnormalities. Too often, people use the concept of mutation to know anything that's wrong with your DNA, including those that are larger chromosomal abnormalities. And to make sure you understand the difference here, you already know what a point mutation is, and that's when you have one of the A, C's, G's, or T's that are out of whack in your DNA so that it could potentially lead to a frame shift or the wrong amino acid being put in place so that the protein that's created is dysfunctional. Okay, so those are point mutations, and they happen to specific bases in the DNA sequence. A chromosomal abnormality are those that are caused by the non-disjunction event that I spoke about in the previous screencast. And the non-disjunction leads to aneuploidy, which is not having the correct number of chromosomes. Now, to be fair, there are two different types of chromosomal abnormalities. So we have those that are caused by a non-disjunction event, but we also have a set of chromosomal abnormalities that are called translocation abnormalities. Now, I'm not even going to talk about these. It's something that if you take a class in human genetics later on, you might talk about those, but I'm going to ignore those for now. Just know that they do exist, but I'm not going to go into the details of those. I just want us to focus in entirely on the chromosomal abnormalities that are the result of non-disjunction. So, as you know, if you have a non-disjunction event, you create gametes, which is sperm or eggs, that do not have the correct number of chromosomes. They are aneuploids. So that when they combine, so let's say we have a sperm that has the wrong number of chromosomes. Let's just say we have this sperm only has 22 instead of 23 chromosomes. And if that were to combine with an egg that has the proper number of haploid chromosomes, then you can see that together there will only be 45 chromosomes in the resulting zygote, which is the first cell that's formed after fertilization. And that's a problem if you don't have enough chromosomes. Obviously, the other issue is if you have too many chromosomes. So in this case, imagine I have, instead of 22, let's say I have 24, one extra chromosome. And I think it's obvious to you that you could imagine that's going to result in having, I should do this in black, 47 total chromosomes. Again, that's going to be a problem in the resulting child that develops. So since we understand the underlying reason for why you would have aneuploidy, because we talked about it last screencast, let's see what it looks like in terms of chromosomes, or what's called a karyotype, of an individual that has the wrong number of chromosomes. Now, you should know that there's a process that I think many of you have heard of called amniocentesis. And amniocentesis is a way of checking before a child comes to full term in the mother's womb. It's a way of checking to see whether or not there are chromosomal abnormalities. And of course, this has to happen before the second trimester, because if you decide that as a result of the chromosomal abnormality you want to terminate the pregnancy, you can do so when it's still legal to terminate the pregnancy. But it's obviously, it's a slightly risky process to go through, and you'll see why. This is what the amniocentesis event looks like. Of course, this looks a little frightening. It's less frightening in reality than it is in this photo. What you're really trying to do here, or the technician who's involved in doing the amniocentesis, he or she needs to withdraw some of what's called the amniotic fluid. And the amniotic fluid is what surrounds the growing embryo here. And know that the growing embryo, it's made up of a lot of cells, but also know that the embryo is going to lose some cells in this process. It's a natural part of the event of growth. Some of the cells are just going to fall off and be floating around inside the amniotic fluid, so that when you withdraw it, you're going to withdraw cells that come from that developing fetus. All right, so you take out that amniotic fluid, and then Here, let me go to another page. You can see here, once you get that fluid out, you are going to put it into a centrifuge, so all the cells go down to the very bottom. And you're going to take those cells and you're going to put them into what's called cell culture. You essentially are going to let them continue to grow on a petri dish. And then you're going to add some chemicals to it to make all those cells stop just at metaphase. So before you start to have the division, this is metaphase and mitosis, mind you. Just before you have cell division occur, where you're going to create two cells from one, you freeze it at that point so that you can see the actual chromosomes. Now this might seem nuts because, as we know, chromosomes are microscopic things. So you need to have a microscope that goes in there and actually takes a picture of all those chromosomes. And then you literally take that photo and you cut them out. Sometimes you do this through, using Photoshop, but in the old days you actually used to use scissors, take a photo and use scissors. You cut out all the chromosomes that are shown in the picture and you start pairing them up. So in the end it looks something like this. It's kind of amazing to see that this is a photograph of the chromosomes because they sort of look exactly like you can imagine they'd look like. Now it might be a little hard for you to see here, but this is actually a pair of homologous chromosomes, and notice that they are duplicates. So this is, if you could just imagine, this is right here. Imagine that's from the mom, and this one's from dad here. And so we have here the centromere in both of these duplicated chromosomes, and it consists of one sister chromatid and another sister chromatid because this hasn't undergone mitosis to separate these things out into individual single-stranded chromosomes. I'm just going to erase this because it gets a little confusing. But you can see what's very important in this whole thing is that the chromosomes all have a very specific banding pattern and length that allows you to be able to recognize that this combination of length and banding pattern is chromosome set number 7, etc. So the largest chromosome is number 1, and the dinkiest chromosomes are number 22. And all of the ones that don't include these two, don't include these two right here, so numbers 1 through 22, we refer to those as somatic chromosomes, and these two down here, the X and Y, are the sex chromosomes. So somatic is 1 through 22, whereas the sex chromosomes here are X and Y. So this is a healthy male because males have one X and one Y chromosome. Let's take a look and see what a healthy female looks like. Okay, you can see here we have the same exact combination. They're a little squigglier, so it's a little harder to see the banding pattern and such, but this is a pretty good technician who knew what he or she was doing. So we have 1 through 22 in terms of the somatic, didn't mean to do that point. Somatic chromosomes 1 right through 22, and then we have 2 X chromosomes for the sex chromosomes, and so that indicates that this is a female because it has 2 X chromosomes. Totally healthy because we have 2 of each, and that's how it should be. You always want to have 2 of each. But of course, if you have an aneuploidy situation, you're going to have either too few chromosomes, you'll have a singlet all by itself, or you're going to have too many chromosomes where you have 3, say, chromosome number 10s, when you should only have 2 chromosome number 10s. So let's take a look at one that many of you already. All right, so in this situation, you can see that, oh, that's funny, I didn't realize that numbers 1 through 5 are missing, but let's pretend number 1 through 5 was just cut off because it's not terribly important. But numbers 6 here through 20 are all paired up correctly, but then number 21, we can see here we have 3 where we should only have 2. So there was a non-disjunction event such that the egg was able to, it was something that brought in 2 copies of chromosome number 21. When you have 3 of something, we call that trisomy. So specifically, this is called trisomy 21, or Down syndrome. And then if you continue to look, you can see here that number 22, it's totally normal, and I think you can see here that this is a male with Down syndrome. Now notice that 21 is a pretty small set of chromosomes here. You should understand that if you had a chromosomal abnormality for chromosomes 1 through 5, which are huge chromosomes, that essentially that's going to lead to the child probably not even making it past the first 2 or 3 cell divisions to create the ball of cells that will eventually become the fetus and then the embryo. So if it's too big, if the chromosome is one of the larger chromosomes, it's pretty catastrophic. So typically, trisomies are going to occur with 18 through 22. You'll have problems. And the sex chromosomes. Okay, so the smaller the chromosome, the greater the chance of having the trisomy lead to the birth of that child. Because if it's 17 all the way up through number 1, the child will never even be born. The child will be miscarried way before it comes to term. So I have this table here because it's sort of an interesting thing to consider. This is when you have a chromosomal abnormality, again due to a nondisjunction, that has to do with your sex chromosomes. So you can see here in this situation, when the child is born with only a single X chromosome and no additional sex chromosome to balance it out, so either no X or a Y. And this is going to be a female, okay, so phenotypically, meaning that expression you will look and see and the person will look like a female, but oftentimes the female will be sterile. But don't forget that in a situation where you have a female, you always have X chromosome inactivation going on, so you really only need one X chromosome. And if this is the case, somebody who has Turner syndrome is going to live a completely normal life in all likelihood and never even know that she has Turner syndrome. She'll just be relatively infertile because half of her eggs will have no X chromosome at all. And if you have no X chromosome, that's going to be a problem because the child won't come to term. And then you can see that we have a whole number of these other ones where in this case there is a nondisjunction, so you have two X's and a Y, and oftentimes people who have Klinefelter syndrome are sort of have giants. They're oftentimes they're giants. In fact, if I'm not mistaken, the commissioner of the NBA right now is somebody who's a Klinefelter's syndrome. I guess it's a hard time to call him a victim because he's certainly a productive member of society. But again, you're going to have potentially some issues with your fertility. And then we can look at this situation where we have double Y, and then we have the trisomy X, and a very unusual 4X syndrome that exists here. These guys are both phenotypically female. This is going to be phenotypically male, also phenotypically male. All right, I guess that's all the time I have for this. And again, let me just bring you back to this original slide that we have here. And this is all chromosomal abnormalities that are the result of nondisjunctions, okay? And make sure you can distinguish between that and a point mutation. Bye.